It's really exciting to be here to tell you about the study entitled The Role of Kinin B1 Receptor and the Effect of Angiotensin 1 Converting Enzyme Inhibition on Acute Gouty Attacks in Rodents. This study was developed by Silva CR and co-workers. Gout is induced by the deposition and crystallization of monosodium urate, and even it can be characterized in 4 well-established stages. Acute attack is the most notable for the agonizing character and the unique pathogenesis of the inflammation and pain it generates. There are some standard treatments available. However, for a growing number of patients, these treatments are ineffective or contraindicated, largely due to the presence of comorbidities. Hypertension is a comorbidity highly prevalent in individuals with gouty arthritis. Interestingly, it was shown that some antihypertensive drugs, as angiotensin-converting enzyme inhibitors, increases the risk of incident gout. ACA blocked the metabolism of several peptides, notably the inactivation of bradykinin. Bradykinin is also a substrate for kinase 1, leading to these are Kinin B1 receptors agonists. Additionally, it is well established that kinins have an important function in inflammatory and painful processes, contributing to some arthritic conditions. The aim was to investigate the possible involvement of B1 receptor in the development of acute gouty attacks, including the one precipitated by ACE inhibitor treatment. Basically, animals were submitted to intra-articular injections, then nociception and edema were analyzed, when appropriate ACA inhibition was done by systemically route. We first defined a low MSU dose, which did not induce pain or edema itself, and a high MSU dose, which was able to induce pain and edema. We observed that both peripheral B1 receptor antagonism with SSR240612, as well as gene deletion by B1 receptor knockout, reduced pain and edema induced by MSU in rodents. Furthermore, B1 receptor antagonism reduced additional inflammatory parameters increased by MSU intraarticular injections, including leukocyte rolling and adherence in blood vessels supplying the ankle. Additionally, animals challenged with MSU had higher articular kinase 1 activity and desired bradykinin synovial levels than animals injected with vehicle. Moreover, we revealed constitutive B1 receptor expression in the articular tissues of rats, and at least in part, these receptors are expressed in sensory neurons projecting from the ankle joint. These neurons are likely to be C and A delta fibers, important for pain regulation. Another exciting observation of our study was that ACA inhibitor treatments induced pain and edema in animals intra-articular injected with the low-dose MSU, which did not induce pain nor edema itself. Articular kinase 1 inhibition and B1 receptor antagonism prevented the inflammation caused by low-dose MSU in ACA inhibitor-treated animals. Low-dose MSU also increased kinase 1 activity and desired bradykinin levels in ACA inhibitor-treated animals, suggesting the involvement of B1 receptor pathways and the increased propensity to develop the acute gouty attack in response to systemically administered ACA inhibitors. Concluding, Kinin B1 receptors contributes to acute gouty attacks, including the ones facilitated by ACA inhibition. Therefore, B1 receptors has therapeutic potential for acute gouty arthritis treatment or prophylaxis, especially in patients taking ACA inhibitors.